Welcome, everyone. This is our demo of Cognistx MoleculeAI platform, which is built to enable drug discovery for small molecule compounds. So just some background on our company. Cognistx was founded in 2015, and we do artificial intelligence work in many different domains, including natural language processing, but also in the drug discovery space. We originally developed the MoleculeAI platform through our engagement with Enveric Biosciences, looking at the properties of psilocybin and similar prodrugs. So some context on the drug discovery pipeline as a whole and where we see MoleculeAI fitting in is in the lead identification and optimization phases. So the entire drug discovery pipeline has many different components, including clinical studies, but we're primarily focusing on the period of time before clinical studies where you're trying to identify which lead compounds are going to be useful for whatever downstream test you're looking for. So if you're looking to design some kind of novel drug that maybe delivers certain benefits to patients in a different way than what's on the market already, you're going to need to look at derivatives of that base compound and try to identify ones that have desirable properties and like low toxicity in the body, for example. So the problems in this phase is that it takes quite a long time. It can take several years. And there are many, many potential lead candidates. So you could be looking at thousands or hundreds of thousands of compounds to try to identify which one is the best. This process is really slow and expensive, not only the assay testing, but also the synthesis of these compounds in the first place. And it requires a lot of data. As a whole, all of these factors make the lead identification and optimization phases quite risky because there's a really high cost of failure. And so our goal with MoleculeAI is to help users focus on compounds that have the best chance of being great drug candidates. And so we see ourselves as de-risking this entire process. So MoleculeAI itself is one centralized platform that's composed of different machine learning components. So one of those components is our high performance predictive modeling framework called AutoMole. Another one is compound clustering and ranking. And then the last two focus on augmenting the synthetic chemist workflow for actually designing these novel drug candidates. So one thing that we really wanted to implement was a continuous learning process with our models. And so the idea is that we can essentially take publicly available data or proprietary data from a client and train a model to understand some kind of property about a compound. So maybe they're looking at the binding affinity or the toxicity of a compound. We can look for data that's available for anyone, and we can train a model to take as input a compound and produce that property as the prediction. We use these predictions to actually reduce testing space. So this is where it actually benefits the drug discovery process. And then once those compounds are actually assayed in real life, we can take those real life results and feed them back into our models so that the models continue to learn on this real world data and hopefully become much more accurate on that particular data set. So if a client is doing an assay that uses one particular hot ligand, we can fine tune our model on their actual results. And ideally, this is going to make that model very customized to that particular client, and it's going to yield much better results than any kind of generalized model possibly could. Our other main machine learning component to MoleculeAI is our AutoMole framework. So this is a fully automated training, hyperparameter tuning, ensembling, and deployment library. And it allows us to go very quickly from a data set to molecular representations, to training many different model architectures and identifying what the best performing model ensemble would be. So the takeaway from this is that it allows us to very quickly automate as much of the data science process as we can. And so this allows us to focus much more on getting feedback from our clients about how our models are performing, and what predictions they might want to see next, as opposed to spending lots of time trying to handcraft different architectures. So for this demo in particular, we're about to look at the dashboard. I just wanted to kind of come up with an example. And so for this case, we're going to look at NSAIDs. So aspirin, ibuprofen, and we're going to try to generate some kind of derivative of these compounds that might have some kind of desirable properties that we're looking for. And we're going to generate these compounds by adding additional substructures to these base compounds, such as rings or sugars at various different atomic positions on these compounds, in the hope that we might be able to identify some kind of maybe a prodrug that could deliver these kind of therapeutic effects in some beneficial way. So with that, we'll jump right into the actual dashboard. And so this is our MoleculeAI dashboard. When you first come onto the dashboard, you're going to be on the All Compounds page. And so this is where we have all of the compounds that are in our library displayed in one easy table. And you can see that we have different metadata about these compounds as well. So you can see where it was uploaded from, the SMILE string representation, when it was added, but you can also scroll over and see different properties. So molecular weight, total polar surface area, number of rotatable bonds. And this is all customizable as well. You can go through and select any of the properties that we have in this dashboard to view as part of the table. And you can also sort the compounds by these properties as well. So we're going to jump right into our clustering and ranking use case. And so in keeping with what we were talking about earlier with the NSAIDs and trying to basically understand which ones we should actually test in the lab. Say we have maybe a limited number of resources. We can only test so many compounds in the lab because it's either too expensive or it would take too long to test all of our maybe hundreds of potential candidates. So in this case, let's look at our NSAID-based compounds and then also the NSAIDs with rings and sugars attached. And so we've added tags to basically identify those in our library. And we are going to generate some clusters. So what this clustering process does is it uses the properties that we've calculated either algorithmically or using our machine learning models to understand which groups of compounds should be clustered together. And this is really useful because instead of us needing to test every single compound in one particular cluster, we can actually only test a subset of some compounds in that cluster. And if we do that and we get our assay results back, we're actually going to have a really good sense of that cluster as a whole. And so we can see this. Here's our clustering results. You can see that we've found 5 different clusters and we've colored them like this. So if we're looking maybe at cluster 0 right here, we can go down, we see some average results, average toxicity, average ADME scores. If we go in and expand this cluster, we can see the individual compounds that make up this cluster. So just clicking on that first one, we see that this is one of our file uploaded compounds, and this is just one of the NSAIDs with an additional substructure on it. We can see some individual scores for that compound, but also we have this flag called representative compound. And so in this clustering, we selected that we wanted to have 5 representative compounds per cluster. And so instead of the 10 to 15 compounds that are in this entire cluster, it actually will suggest that we should actually test this compound and a couple other ones in this cluster. And if those are the only ones that we have the resources to test, we're still going to understand the properties about all of these compounds in general. And so this can be really useful when you're trying to understand maybe the toxicity properties of an entire cluster. So if we test a couple of these compounds and we see that those couple are very toxic, it's much more likely that these compounds altogether are going to be toxic than, say, a compound coming from one of these other clusters over here. And actually, right now we're looking at all of the properties, but we could actually even focus in further by looking at only the toxicity clusters. So now it will only take our toxicity properties into account when we actually perform this clustering. And we'll talk about this a little bit later, but there's also additional kind of granular properties associated with toxicity as well. And we could even go more in depth here, but we'll just kind of leave it as overall toxicity and let's try to regenerate those clusters. So in this case, the only properties that are actually going to influence the way that these compounds are being clustered is the toxicity properties. And so we see, oh, we've generated 5 new clusters, and these ones certainly look different. We see this one cluster up here that's actually only a single compound, which suggests that maybe we want to look at that one a little bit closer. But we can also see that maybe cluster 4 down here has a overall average toxicity score of 43. And so this one seems to have a relatively low toxicity score, which in this case, because our scores are normalized, is actually meaning this class of compounds right here is more likely to be toxic, or at least has similar toxicity properties. So let's jump right into one of these individual compounds right here, and we'll see some more information on that compound in particular. So this is one of our NSAID-based compounds with a ring or sugar attached. And we can see this radar chart here that gives us an overview of the properties for this compound. So these are all normalized scores and they're customizable. Right now we have 6 different categories of scores that we're looking at: ADME, toxicity, binding affinity, synthesizability, novelty, and prodrug likeness. And so all of these might be useful depending on what kind of downstream tasks or therapeutic effects you're looking for. So in this next pane, we have score details. So this gives us the actual granular metrics about this compound. So we have some general properties on the left, but on the right side, we can also select one of these larger compound categories like toxicity, and we can actually see all of the toxicity related metrics for that compound. Below, we also have similar compounds. And so if you are trying to design a novel compound candidate, a lot of times it's really important to understand how similar that compound is to existing compounds that are already on the market. And this also might be useful for protecting intellectual property. So if you're looking at certain synthesis pathways for these compounds, you might be interested in, say, like the related records of these kind of compounds that might be similar. You might be interested to see if there are any patents that cover certain pathways of these kinds of similar compounds. In this case, here's the compound that it found that it thinks is slightly similar to the one that we're looking at, but the similarity score is 0.9, so it could certainly be higher still. On the bottom, we also have an area where users using the tool can add notes for these compounds. So say if they thought that based on these properties for this particular compound, they thought that this compound had a very good chance of being maybe a desirable compound for like the next phase of drug discovery. Maybe this is something that we should consider for clinical testing, or we should do much further assay testing on. You could write that information in this notes field, and you could also add a user prediction. So on this right-hand side here, you see low, medium, and high. These are essentially likelihood scores that a user could give to this compound, saying that they either think it has a high likelihood of being able to go through to the next phase, or maybe it has a low likelihood. They think that this compound is maybe too toxic or doesn't have the desirable properties that we're looking for. And as a result, we should focus on other compounds first. So the last useful tool for a synthetic chemist is going to be to actually try to make modifications to this compound. Because again, we're looking for derivatives of our base compounds that might have more desirable therapeutic effects. And so if we click this Create New Compound page, it's actually going to load up our Create Compound page and preload that compound into our molecular editor. So this allows us to actually make structural modifications to these compounds and visualize how those modifications affect the properties. So for the sake of this example, let's assume that we're looking at the inhibitory constant value for the 5-HT2A serotonin receptor. So for this particular compound here, let's just leave it as is, and we'll say that we're trying to minimize that particular property. So we can go ahead and generate our contribution map. And so this is essentially a way for us to view the properties associated with the compound that's presently in the editor, but also visualize the actual atomic contributions to that property. So if there are certain substructures of this original compound which contribute more or less towards that 5-HT2A value, it's actually going to highlight those as well. And so we see these highlights here on these particular atomic positions. So we see that for this compound, with no modifications, our 5-HT2A value is 4.2. So let's say that we're really looking for a compound that actually has a much greater binding affinity towards the serotonin receptor. We really think that we'd rather it be somewhere closer to say 1. So let's try to make some kind of modification. Let's take this carbon right here, and let's just add a hydroxyl group. So that's just one little modification, but I'm curious, how is that going to affect our properties? So with this modified compound, we can go ahead and regenerate our contribution map, and we'll actually be able to visualize how this structural modification affects the actual properties of this modified compound. Cool. So I think our original score was 4.22. So actually this one has a 5-HT2A predicted score of 4.27. And so in this case, we actually think that adding this hydroxyl group might have a detriment to our overall score. If we're really trying to minimize it, then maybe we would want to make some kind of other structural modification. So on the right-hand side, we can also see a compound history. So this just allows us to not only view the most recent contribution map and the properties associated with that compound, but also a history of all of the contribution maps and all of the modifications that we made. And so this could be really useful if you made many different modifications and you wanted to go back and see which one had the best value for that property of interest. Or maybe you were just curious how kind of the trend, what the trend of that property looked like as you made certain types of modifications, maybe starting from multiple different base compounds. And this also aligns similarly with our other generation methods. So right now we're just looking at one individual mapping. So this is just one compound generating one mapping, but we also have some more automated ways to do this. So for substructure additions, this allows you to actually select some substructures. So these could be like the rings and sugars that we added to our base compounds. And these substructures will be automatically added to various atomic positions on that compound. So maybe we know that atomic position 2, where we added the hydroxyl, maybe that's a really good position for us to add additional substructures onto. So what we can do is we can select various different substructures, and we can go ahead and put in atomic number 2, and we can also visualize those exact same properties that we were looking at earlier. And we can generate the mapping, and it will automatically add all of those substructures on to that atomic position. And so this is one way, instead of having to manually go in and add each of these structures by hand, you can automatically add them, and it will generate the properties for all of them. And then afterwards, you can go through and you can see which of those substructures helped me the most in terms of these compounds have much more desirable properties, adding this particular sugar at that atomic position. And you can actually specify multiple atomic positions if you're looking at multiple different areas of the compound for modifications. Our last one is slightly more experimental, which is expanded space. And so this is similar to the substructure additions, but this is actually an automatic process. So you're starting from this base compound, and maybe we're looking at, again, the 5-HT2A inhibitory constant value. But this time we're going to automatically generate similar compounds to this base compound. And so this flag right here allows us to choose how many compounds we'd like to generate. And this control right here lets us set kind of a similarity threshold. So if we want all of our compounds to be at least 80% similar to our base compound, then we can go ahead and just click generate mapping, and it's going to automatically create similar compounds that are valid in terms of their chemistry, and we'll be able to see their properties as well. So this is one other way of kind of aiding the process of a synthetic chemist when they're designing novel compounds, where we might be able to give them some kind of inspiration through this automatic compound generation. So that's the end of our demo. If you have any other questions about MoleculeAI, feel free to email me at justin@cognistx.com, justin@cognistx.com, and I'd be happy to answer any of your questions. Thanks.